Welcome back to Botany class. In the previous class, we discussed a detailed account on Ploidy or numerical chromosomal aberrations. You know there are two different types of chromosomal aberration based on structure and the number. Earlier, we discussed about the numerical chromosomal aberration. What are the changes takes place due to number, that is ploidy. Today, let's start with structural changes in chromosome. There are some structural changes takes place within the chromosome segment, that is otherwise called structural chromosomal aberrations. The variations or changes are known as aberrations. Within the normal DNA segment, variations takes place either by arrangement of the genes or by the number of the genes. Due to this, there are different types. Based on this category, this chromosomal aberrations are classified under two headings. Based on the breakage and reunion. What happens actually when the chromosomal aberrations, structural chromosomal aberrations takes place? The normal segment of chromosome breaks and reunion of genes taking place. This is due to ionizing radiations or chemical compounds. Either by ionizing radiation, you know that mutation takes place by means of ionizing radiations or chemical compounds. Due to this, these structural aberrations takes place. Based on this breakage and reunion, there are two different types of structural chromosomal aberrations. They are based on the number of gene loci or loci and the arrangement of gene loci. Arrangement of gene loci. You can say loci or loci. Number, either by number of gene loci or arrangement of gene loci. Look at this diagram. This is the normal chromosomal segment. So, this structural changes occurs by means of addition or deletion, or either deletion or duplication. Duplication of some extra segments or deleted some segments from the normal one. That is this gene number and arrangement, either by the structural changes occurs by number of gene loci. In the particular number, normal number, how many genes are located, there will be a variation if it deleted some segments, or the arrangement, modifications in the arrangement of gene loci. Depend upon this, there are four different types of this structural aberrations. Based on this number, it's classified as deletion and duplication. Deletion and duplication. This deletion is otherwise called deficiency. If anything deleted in this segment, the characters, if some genes are deleted within a particular normal chromosome, it causes deficiency. Some characters loss of some characters. And duplication is otherwise called repeat. Deletion is deficiency, which causes deficiency. Duplication, which causes repetition. The same genes are repeated again and again, few segments, that is duplication. So, based on the number of gene loci, there are two different types of chromosomal aberration. Then based on this arrangement of gene loci, there are two types, inversion and translocation. Here there are two different types, inversion, translocation. Let's see one by one. What is meant by deletion? So at first you know about the classification. Structural chromosomal aberrations are classified under two categories. They are based on the number of gene loci and the arrangement of gene loci. Look at this diagram. This is the normal chromosome segment. Normal chromosome, which contains, the chromosome contains gene locus. What is meant by locus? The genes are located in a particular position. So A, B, C, D, E, F, G, H and I. There are number of genes are located in a particular locus. A located in this locus, B located in this locus. So this is a normal segment. In the normal segment, variations occurs. Variation take place. Deletion. What's meant by deletion? Loss of chromosomal segments. The deleted, some part, some portions of the chromosome is lost. That is called deletion or deficiency. So this deficiency are two different types. They are terminal, that is this classification based on the position of the deletion, where the portions of gene deleted or lost, or when this, where the segments are breaks, that lost its gene is called deletion. So two different type based on the position, terminal deletion and intercalary deletion. So here there are two different types, terminal deletion and intercalary deletion. You know that what is terminal? Terminal is the tip of the chromosome. So here there is a tip of the chromosome. This is the example of terminal deletion. Look at this segment. Here A is lost. This is the normal segment. Here in this segment it starts from B, C, D, E. All the remaining genes are located there. Remaining locus are present there. But terminal part, the A is missing. So that is terminal deletion. Intercalary, in between the segment, the part of gene or the portions of the chromosome in between the segments. For example, here A is present, B, C is present, but this D is missing. So where this intercalary, this type of deletion is known as intercalary deletion. So deletion, there are two different types. What are they? Terminal deletion and intercalary deletion. Then another type of this chromosomal aberration based on number of gene locus is duplication. What's meant by duplication? Repetition, repeated. Some segments or some portions of the chromosome repeated. Example, here there is this is the normal segment. The duplication, some segments are repeated. So this all observed in Drosophila and maize. These two different types of deletion and duplication, it's observed in Drosophila and maize. Examples are very important for one-word questions. Now duplication, the segments or the portions of chromosomes which repeated once again. Look at this. This is the normal segment which contains A, B, C, D, E, F, G, H and I. But in this A, B and C are repeated again. B and C segment. Here one B, C is present. One more, once again the B and C is there. So this is aberration. You know that each and every organisms which possess a definite number of chromosomes and the genes are located in a particular locus. It should not be vary. Here the variations occurs. So it is aberration. So in this B and C is repeated. So it is called duplication. And this is the tandem B and C. In the normal position, once again it repeated. But in this, the B and C, the order it repeated. Here B and C, again C and B. So this is reverse translocation. And this is reverse duplication, not translocation, it's duplication. Duplicated, B and C duplicated, but the order in the first segment B and C, once again B and C. Now in the second segment B and C, again C and B, the order also changed. Then displaced, sometimes the particular gene displaced its position. See here A, B and C present, again D, E, F, after G once again B and C is located. So this type of duplication, these all different types or duplication comes under duplication. So these all number based on its number, the particular number of gene which is duplicated once again, that is repeated once again. Example, Drosophila and maize. Then second type of classification is the arrangement, based on the arrangement of gene locus. These all based on the number, now based on the arrangement. During this arrangement, it is classified into inversion and translocation. So in your book, translocation is given. So translocation, the transfer of chromosomal segment to a non-homologous chromosome. You remind this word, non-homologous chromosome. Translocation, transfer of DNA segment or transfer of gene or chromosomal segment to the non-homologous. Non-homologous is the heterozygous condition. You know, already you learned about homologous, non-homologous. So there is you should not confuse it with crossing over. In crossing over, there is a difference between crossing over and translocation. In crossing over, the homologous chromosome, the changes, the segments here. In crossing over, what happens? Homologous chromosomes, the segments are exchanged within the homologous chromosomes. This is homologous chromosome which obtained from parents, both the parents. So this segment exchange, if A and B, capital A, capital B, capital A, capital B, here small a, small b, small a, small b. In this, these segments are exchanged. Chromosomal segments exchange. But here, the segments exchange non-homologous. So different. It exchanged in different manner, non-homologous chromosomes. So in this crossing over, this segment, homologous chromosomes exchange its segments, this and this. This is the tetrad, this is a tetrad. These two segments are homologous chromosomes which exchange its segments in crossing over. But in translocation, it exchanges within non-homologous chromosomes. This point you remember it. Non-homologous translocation, transfer of segments in the chromosome, transfer of segments or the gene to non-homologous chromosomes. So this is the classification, the structural chromosomal aberration. Second question you are going to learn today is DNA metabolism. DNA metabolism. DNA metabolism. As the repository of genetic information. Repository means it is a place where the things are located. So, repository of gene. So, DNA which occupies a unique and central region of metabolic or biological macromolecule. So, it is unique. DNA is unique. Here metabolism of DNA. The term metabolism includes three different processes. They are replication, that is its exact copy is duplicated, replication, synthesized, newly synthesized, replication of DNA. So, metabolism is the process which includes replication, repair and recombination. So, DNA metabolism is the process which includes replication, repair and recombination. And DNA is unique and it's the marvelous device which stores the genetic information. So, DNA is the biological and it is a macromolecule which is marvelous device. Why? It stores the genetic information. All the genetic informations are carried within the DNA. So, the term DNA metabolism, this term DNA metabolism which includes or it describes these three different processes. What are they? Replication. What's meant by replication? The DNA duplicates. How it duplicates? There is a process in plants, this replication of DNA which duplicated, it newly synthesized, the exact copy of DNA is synthesized. Let us see how it replicates. And second process is known as repair. So, there are some damages in the plants, but these damages are rectified by means of this repair mechanism of DNA. So, that DNA is unique in nature because it restores all these problems or solve all these problems. Then recombination. So, this DNA metabolism includes all these three processes. Let us see what is replication, how replication taking place. So, replication is the process by which the DNA is synthesized its exact copy. You know the DNA which is double stranded helical structure, double stranded helix. So, this helical structure, so it starts from 3 dash 5 dash 5 dash 3 dash direction. So, it is the double helix of DNA. During replication, what happens? This DNA, the parental strand, in the double helix, this is the double helical structure, the parental strands unwind and a newly synthesized strand. A strand is synthesized newly. So, both the strands are synthesized. So, this is replication. So, during replication, in this type of replication, in eukaryotic replication is semi-conservative type. The type is represented as semi-conservative. Semi-conservative means semi, half conservative. Conserve means its protection. So, semi-conservative method or discontinuous method. When it is synthesized, it is synthesized as a discontinuous nature. So, how it is synthesized? So, there are some enzymes involved in this process. What are the enzymes? There are helicase enzyme, topoisomerase, polymerase and ligase. This polymerase, there are three different types: polymerase, alpha polymerase, delta polymerase and epsilase. So, these three different types of polymerase enzymes are involved. At first, you know about these enzymes and its functions. These functions of enzymes are very, very important. 2-mark question or 3-mark question, it is very important. What's the function of these enzymes? So, first question, 2-mark question or 3-mark question, what are the enzymes involved in the replication of DNA or DNA duplication? So, when DNA replication, this double helical structure starts to unwind. So, this is a double helical structure, it starts to unwind. When it unwinds, this is the portion. So, origin, there are different origins are there, it starts from a particular origin. So, in this origin, it started to unwind the chromosomal segment, the DNA segment. It is a coiled structure, you know very well. This coiled structure started to unwind. So, this unwinding, for unwinding this helicase enzyme is important. So, function of helicase enzyme is which unwinds the DNA strand, double strand. Due to this, here there is a fork formed. This fork is called as replication fork. The fork is known as replication fork. So, in this replication fork, just above this, so this here it started to unwind. Above this, these are supercoils. So, it coils tightly. So, this are called as positive supercoils. Once it started to unwind or once the replication fork is formed, the remaining part above is started to coil, that is called positive supercoil. So, this positive supercoil can be removed by another enzyme called topoisomerase. So, the function of this topoisomerase enzyme which removes the supercoil. If it is coiled tightly, it cannot unwind. So, the enzyme involved there, topoisomerase, which removes the supercoils. So, diagrammatically it can also represented by this. Here is a replication fork and this is one strand, this is the another strand. Here 3 dash 5 dash and this direction 3 dash 5 dash direction and this part here how it forms. Let us see. This is the replication fork, this region replication fork, and this replication fork here is supercoils which is removed by this enzyme topoisomerase, and helicase, which here is the enzyme helicase which unwinds. Helicase, this is topoisomerase enzyme, and so here it is 3 dash 5 dash direction, here 3 dash 5 dash direction. two different strands. This is the replication fork. Now, the newly synthesized strand from this opposite direction, from here the newly synthesized strand is formed. So, this newly synthesized strand is formed and this strands involved the enzyme polymerase enzyme, which extends the strand, DNA strand. Extend the DNA strand towards the replication fork. And this polymerase enzyme alpha, delta and this 3 are involved. This alpha polymerase enzyme alpha, which synthesize a short RNA primer. The DNA, newly segmented DNA are synthesized in a bit fragment, small bits. It forms as a small bit. This is called as Okazaki fragment. As a small bit, Okazaki fragment. So, it forms as a small bit. This is semi-conservative method and this is elongated by means of, these strands are extended by means of DNA polymerase enzyme. So, this enzyme, this third type of enzyme which extends the DNA polymerase enzyme, this one. And the newly synthesized strands are formed by means of RNA primer. So, this primer enzyme which discontinuously it formed and it is pasted by an enzyme called ligase. Here there is an enzyme ligase which pasted this Okazaki fragments and leads. So, in one end it is lagging strand, another end is leading strand. So, it extends the polymerase enzyme in the leading strand, another side it is lagging strand because it is formed as a Okazaki fragments. So, these Okazaki fragments are pasted by means of the enzyme. What enzyme? This ligase enzyme. So, the role of these enzymes are very, very important. What are the different types of enzyme involved in replication? That is helicase, topoisomerase, polymerase enzyme. 1, 2, 3. There are 3 different types of polymerase enzyme which are named different types. So, this is replication. So, newly synthesized. As a result, it forms a new strand DNA. The exact copy of DNA is synthesized. So, it is called semi-conservative method. As a discontinuous, it synthesized and it is elongates or it is joint or it is extend the strand. So, DNA replication. This is DNA, one part of DNA metabolism. And second part is repair. What is another role of this DNA? DNA is unique because there are number of damages taking place due to ionizing or chemical compounds, some damages. So, these damages are caused. You know that the plants are sessile. The plants sessile means it cannot move place to place. The plant is fixed in a particular place. How do they protect themselves from the exposure of sunlight? So, during the day, it is completely exposed, the plants, animals which can move here and there to escape from the sunlight, and it gets shelter under the tree. But the trees cannot move anywhere because it's a sessile plant. It fixed, sessile means it fixed in a particular place, it cannot move, immobile. So, there are some mechanisms, DNA repairing mechanism. It synthesizes some enzymes. There is an enzyme called photolyase enzyme. So, this photolyase enzyme which repair the thymine dimers. The thymine dimers or repair. So, repairing mechanism by means of this DNA which synthesizes some enzyme. So, the thymine dimers restores the DNA, structure of the DNA. The structure of the DNA without causing any damage, it can restore. This is one of the process. Then, the main role of this DNA repairing mechanism is it maintains the genome or genetic integrity. It maintains the genetic integrity, and due to this it recognizes the mutation and the DNA segment. Either mutation takes place or there is no mutation, which recognizes and removes the mutations from it. That is one of the DNA repairing mechanism. How is the genomic stability maintained in living organisms? You know, this chromosomal numbers, if any changes takes place, it leads to mutation. So, these mutations cannot be affect the metabolic process. There are number of processes taking place within the plants. It cannot be affected. So, this mechanism, this repairing DNA mechanism, DNA repairing mechanisms are used, are very important, or which is playing very important role by this type of damages. So, one thing, it protects from the exposure of sunlight. So, this enzyme, photolyase enzyme, and this, why this DNA is unique? Because it's the only macromolecule where it exists the repairing system. DNA metabolism contains a system of repairing mechanism. So, it is very important. And this DNA, which is subjected to various types of damaging reactions. The various types of damaging reactions it restores. It restores all the different types of damaging processes. Some damaging processes takes place within the plants itself, which can restore by means of this DNA metabolism. So, first, DNA metabolism is used for, or it plays important role in replication process. Then it is restoring the repairing mechanism. And third one, recombination. Already you learned about recombination. As a result of this crossing over, the segments are exchanged and it forms the recombinant gene. So, the new varieties are formed. For example, if these segments are exchanged, what is the result of this? First, this first and last, it resembles to the parent, which contains this A and B, and the second one, which is the recombinant one, which possesses half of this, another half from the neighboring segment. So, this is mutated. This capital A is present, and here it comes the small b. So, next segment, it consists of this small a and it exchanges this capital B. So, this is exchanged here, this one. So, these two are recombinant, and final segment, which is A and B, small a, small b. So, among these four, these two are recombinant segment. So, recombinant segments are formed. So, recombination is a result of crossing over and it leads a new variety. New variety is formed. So, DNA metabolism includes, or DNA metabolism describes these three processes: replication, repair mechanism and recombination mechanism. So, in this, up to this, we are going to stop the third lesson, because biology students, in the biology book, you have this much topics, but science group students, pure botany, 2 more topics are there, 3.6 and 3.7. So, at first we are going to study the common topics among these 2 books. Later we are going to study the extra topics present in the pure botany book. And most probably next week or this month, the last week, you will have the one word test. So, you are going to prepare first 3 lessons. Science group students, that is pure botany, you finish up to this question, DNA metabolism. 2 more topics, let us complete later. And biology students, you complete the first 3 lessons. Today on, you start to prepare, and the questions which do not contain multiple choices. So, you yourself, you enjoy learning by reading and preparing the question. It is not new to you. So, I hope that you maintain the class note, completing all the diagrams. Likewise, you start to read page by page and make it as a hints. Prepare yourself as a one word question. So, everybody start to prepare one word question and prepare well for the coming exams. If you are writing it once, you recall everything once you are reading. So, main thing, you must read the book again and again. So, first lesson, in the first chapter, there are number of examples are there, one word questions. There are many one word questions in the nook and corner. In the corner, some do you know part is there. Do not leave that. Every part, every line you read very well and make yourself and maintain the one word questions and learn it again. Before the test, you learn once all the 3 chapters, complete the one word question and write the test. All the best.